Hi, everyone. My name is Nikki Traylor-Knowles. I'm an assistant professor of marine biology and ecology at the University of Miami Rosenstiel School of Marine and Atmospheric Sciences. And today I'm going to talk about some of the work within the field of ecoimmunity that my laboratory has been doing along with collaborators to develop and understand the cellular mechanics of corals to be able to develop cellular assays to inform restoration. So if you're interested in connecting, here are my Twitter handles and my email, and also our laboratory website. Our laboratory is located here in Virginia Key right outside of Miami. And this is my laboratory here. And I just want to give a particular shout out to Grace Snyder, who is the graduate student who has been leading a lot of the work that I will be presenting today. So one of the biggest questions that has always driven my work is, why do some corals survive and some don't? And I think that one of the primary answers to this is the coral immune system. Corals have the ability, have actually a really complex immune system. And unfortunately, I don't have time to go into this, but the areas that my laboratory has really been focused on are understanding immune genes and pathways that are important for coral immune system function, as well as the cellular aspects, and in particular the immune cells, but also more recently stem cells and their importance into coral survivability. And as we all know, corals are also holobionts, and so that adds sort of another layer of complexity to their immune system. So they have to have an ability to be able to understand what is self versus non-self on a lot of different levels because of so many microorganisms that are coexisting with them within their gastroderm and epithelia. So we have a lot of clinical tools that we use for assessing coral health, from histology, where we want to understand the tissue structure, to omics, we're understanding, a broad scale understanding of their genetics and gene expression, or protein expression, or metabolic expression, to enzymatic assays, in this case, looking at phenol oxidase activity in different corals, to now recently, what my laboratory has and collaborators have been working on is developing flow cytometry to look at live cells and be able to use that as an assay to, again, contribute to all the other tools that we're already using to assay and understand the health of corals. So flow cytometry is, or fluorescence activated cell sorting, is a process in which you can use different lasers to identify the cellular aspects, or the cellular aspects of different cells. So from granularity to size and shape, to whatever their fluorescent output is. And so it's a way in which that we can separate out different cell types and then actually collect them live, and then we can start doing assays with them. And I just want to again recognize my graduate student, Grace Snyder, and our collaborator, Benjamin Rosenthal at Ben Gurion University in Israel, that we're working with and have been working with on using this technique. And so coral cell biology is really one of the least understood of coral immunity. And that's because it's really challenging. Like I mentioned before, there's a lot of microorganisms and other things that live within corals, and being able to do cell culture with this has been a huge challenge. And so using FACS, we're able to isolate out different cell types and then start to really characterize them, right? Whether it be through sequencing or through cell culture assays, or histology, or other types of assays that we can then just focus on one specific type of cell. And so this is an immune cell, for example, that we've been able to isolate. It's engulfed. You can see the two green beads that's engulfed. You can see the lammopodia that are reaching out, searching its environment, looking for more things to engulf. And additionally, the other cell type that we've been really focused on are putative stem cells. And so I'll talk about some of this work now that we've been doing in the context of how we're applying it. And again, this is the work done by Grace Nader. And so why we're focused on this, on cells in particular, is that we need tools for assays for immune health. And we need a toolkit of strategies to help increase temperature or disease or whatever tolerance within corals. And so while folks are putting corals out on reefs, we also need to understand and figure out ways in which we can help those corals survive when they are on the reef. And so that's where this is leading, although we're in the very early stages of it. And so the question is, how do we apply this work that I've been discussing? So for example, we can use the discovery of the immune cells and their ability to phagocytose or to engulf foreign particles as a measurement for their immune reactivity. And so in this figure here, what you're seeing is this bar here represents a diseased piece of A. cervicornis that has white band disease. This is the margin near the disease, and then this is the healthy piece. And what I want you to appreciate is how much higher this piece, this bar is, indicating that within the diseased tissue, it's phagocytosing more. So indicating that there is an immune response that's occurring, and that we can see and show that there is an actual immune activation that's occurring, and that this coral is indeed fully sick. Additionally, we've started to apply this to stony coral tissue loss disease, and where we're starting to understand and look at, a lot of times we get a piece that will have presumably healthy tissue next to a disease margin. And so we wanted to test, is this healthy part actually healthy? How does it compare to the margin when we look at its ability to phagocytose? And so we compared that to a control as well as a piece that had recovered after a week. And what we saw is that the healthy and the margin were basically the same. There was no statistical difference in terms of their activation of the immune response. And this is compared to the recovered and the control, where you can see it's much lower. So again, indicating that despite the fact that it doesn't look sick, it is sick. And this, I think, is very, has the potential to be really useful for our understandings of coral health. Now, of course, we're at the beginning of this where we need to start actually figuring out the foundations of everything. But the applications here, I believe, are really, the potential is big. And additionally, we've been working on with the idea of being able to do stem cell transplants. And so the idea is that we can take cells, macerate them, run them on the flow cytometer, isolate out different cell populations, and then sequence it, and we can get genetic information about each of the cell types that then we can apply to being able to actually better isolate more purified cells. And in the case of stem cells, the idea is that then we can be able to isolate stem cells from either a more temperature tolerant or disease tolerant coral and transplant them into a coral that is less tolerant. And so we're, again, in the beginning stages of this, and we're using different models, including anemones, to be able to do this work and to eventually apply it to corals. But our hope is that we could have this as a tool, much like a bone marrow transplant in humans, to be able to use that within corals. And so I just want to close that, we're really at a frontiers of coral immune cell mechanisms, where there's a lot of things we don't know and a lot of things that we're starting to understand. And I think it's really exciting. So we're developing a lot of live cell assays to disentangle the immune system health and function in health and in disease. Understanding the connection between immunity and stem cells and cellular reprogramming, which I think will be really critical for understanding of coral health and coral recovery. Develop functional assays for understanding the gene mechanisms within corals, and dissect the holobiont system in live cells. So identify the recognition system. So there's a lot of things that we're working on and excited about. I think that there's a lot of potential for this type of work to contribute to all the other amazing things that are already going on in restoration efforts to understand coral health and repair reefs. Lastly, I just want to give a shout out. If anybody here on this recording identifies as a Black woman in ecology, evolution, or marine science, please connect in our group BWEAMS. It's free. And so you can sign up on our website. You can also connect online. But we'd love to have you join and help build our community. And so with that, I would like to thank my collaborators, in particular, the folks in my laboratory and Grace Snyder, who has really led this work, and our collaborator, Benjamin Rosenthal, and his laboratory for their help with and assistance collaborating with a lot of the stem cell and immune cell work. And then the folks, Val Paul and Erin Muller and her team, and Blake Ushijima, for their help with the stony coral tissue loss disease work that I briefly alluded to in this talk. Thank you so much.